As you know, I am a senior research fellow in UCD Ireland. So today I'll be talking about the redox biology and innate immunity in the mucosal immune system, particularly in gastrointestinal tract. I also work on pulmonary tract, but today, just for the sake of simplicity, I'm going to present just for the um, intestinal immune system. So before we go into details, I'm going to just show few brief slides of how our intestinal immune system and what different components they have. So obviously the intestinal immune system or the mucosa is the first line of defense because we know we harbor trillions of microbiota, bacteria, fungi, helminths, viruses in our gut lumen. So obviously the epithelial layer or the mucosa is the primary defense, physical defense barrier between outside environment and our body. And they maintain this balance as homeostasis. And combined together, the gastrointestinal mucosa is one of the largest immune system of our body. And when we talk about the components, what we call at gut-associated lymphoid tissue. So this is a mixture of all innate immune cells and mucosal immune cells, which combine together to form innate immunity. And they are structurally can be named as PS patches, so which harbors. One second, I'm going to just show a pointer. So these are the PPS patches, so which harbors all innate immune cells, dendritic cells, T cells, macrophages, neutrophils. And this is the epithelium where we have a specialized M cells where the dendritic cells sample these and then sample antigens from the lumen. And then there are several aspects of tolerance and anti-inflammatory or inflammatory response is mounted. So our intestinal mucosa does two things. One is tolerance, because of course, if we harbor trillions of bacteria, we can't be um, mounting anti-inflammatory or pro-inflammatory response all the time. So there's always a tolerance. So the body understands, so these bacteria are commensal. I'm not going to mount any response. I'm not going to do inflammation. But as soon as there are pathogens or antigens associated with pathogens, these immune systems are equipped with specialized mechanisms where they can identify these pathogens and then mount a response. As I said, these epithelial barrier, they recognizes they have toll-like receptors and they recognizes patterns, antigens. They also form a physical barrier, so and there is a tight junctions as well, so there is no um, um, unwanted movement of luminal microbiome to the host, so that it maintains its tolerance. And the epithelial cells, they secrete a variety of factors. They secrete antimicrobial peptides. They also secrete mucus, goblet cells, which forms a physical barrier just outside of the epithelium. They also secrete cytokines and many other immune modulators. And one of the secretory product of these epithelial cells are a reactive oxygen species, which is the center of the talk today. So I'll be talking more about reactive oxygen species. Just before that, just would like to give a brief introduction. And these secretory products, then they interact themselves, or the secreted products secreted by the epithelial cells then interact with immune cells, variety of immune cells, T cells, macrophages, residential dendritic cells, and when there is inflammation, either with neutrophils as well. Now, when we talk about reactive oxygen species, uh, in the mammalian gut, mammalian intestine, or any other mammalian system, uh, the redox is reduction and oxidation. So it combines oxidants as well as antioxidants. So this is the study of the whole system, generation and degradation of oxidants. So when we look at the oxidants, the most and primary enzyme which makes reactive oxygen species, which is a deliberate, are NADPH oxidases. So I'm going to talk this about in few slides later. But just for you to remember that the most of reactive oxygen species which are made in our body are actually byproducts of many other enzymatic reactions. But NADPH oxidase are designed to make reactive oxygen species. So reactive oxygen species made by these enzymes are not a byproduct. They are the main product of the enzymatic reactions. There are reactive oxygen species, of course, secreted by mitochondria in normal as well, because we know there is a leaky electrotransport chain sometimes. And when there is a stress, the mitochondria makes more ROS. There are nitric oxide synthase, which makes nitric oxide as well as peroxynitrates. And this in general broadly combines what oxidants we deal in a mammalian gut. Now, in parallel, there are, of course, antioxidant systems like catalase, superoxide dismutase, and glutathione peroxidase. And these antioxidant systems provide a balance to the production of these oxidants so that the production and the elimination of these oxidants are maintained. And when this production or this balance is stripped off, for example, in case of inflammation, as you can see here on the right, this is a mouse intestine. So at homeostasis, this is what generally you look at it in hematoxylin, eosin staining. And when there is inflammation, you see lots of these blue dots. There is thickening of mucosa, thickening of muscularis layer. And this is particularly hallmark of inflammation, what you see. And most of these inflammations are actually orchestrated by as well epithelial cells, but also in here innate immune cells, primarily macrophages and neutrophils. And these cells, they make lots and lots of reactive oxygen species. Now, I will go in detail what type of reactive oxygen species, because reactive oxygen species or ROS is just umbrella term. So there are many more when we go in detail, different types of ROSes. And different types of ROSes have different type of functions, different type of half-life and what they do. So, but in general, they make rich oxygen species, and that causes inflammation and damage. And mostly this is what we see in colitis model, or which is a mouse model of inflammatory bowel disease. So there is always a balance between oxidant and antioxidant system in the gut. Now, how these ROS production is regulated. So we'll just go a brief and this is what we mostly looked at in the previous slide. There are NADPH oxidase system. There is mitochondrial respiratory and respiratory burst. There are many other oxidases in the cytoplasm as well. Together with any exogenous source such as pollutants, radiations, or any unwanted chemicals, they make a reactive oxygen species. Now, in general, the ROS production is very beneficial because at low level of ROS production, this reactive oxygen species, they act as a secondary messenger. So within the cell, they go and interact with several proteins, enzymes, transcription factors, and they together regulate cell proliferation and differentiation. That is very important for the gut epithelial renewal, because remember, the gut epithelium renews every 72 to 75 hours. So the stem cells in the gut epithelium has to renew fast, and that is maintained at homeostasis by a lot of secondary messengers, and one of that is reactive oxygen species. ROS as a secondary messenger also play a response in immune function, as because it is a secondary messenger, it plays a response in signal transductions, apoptosis, and autophagy. Now, when this ROS production is by any means either endogenous or exogenous in increased increased to a level than normal, we call it high ROS. And at the same at that point of time, of course, because the antioxidant systems are not catching up, there is an impaired antioxidant system. So you're making more ROS as compared to you are degrading ROS. So there is high accumulation of these reactive oxygen species within the cell, outside the cell. They create oxidative stress, and eventually, because they can go and react with a number of enzymes, even DNA, they make damages to lipids, proteins, and our DNA, and eventually may also cause to cell death. Now, if we go a little deeper, this is detailed how these functions play around. So NOx is, in short, what we call for NADPH oxidase. So I'm going to talk this about this in much more detail. But there are endogenous sources, peroxisomes, mitochondria, and endoplasmic reticulum as well. They make reactive oxygen species, and exosomes, as I said, extracellular, sorry, extracellular source. And primarily, what we talk about is mostly two types of reactive oxygen species. One is superoxide. So this is oxygen stripped of electron, and another is hydrogen peroxide. Now, these are two main primary reactive oxygen species in the gut. Now, at physiological levels, at here, when they are produced low, they, as I said, they act as a secondary messenger. So they go and modify enzymes, iron sulfur modification of enzymes, or cysteine modification in the enzymes, and several many enzymes. And they then go activate several signaling pathway, what we term combinedly called as redox signaling pathways. And these different proteins, enzymes, transcription factors, then they play role in immunity, proliferation, development, and many other physiological roles, and that they are very essential and very important for our day-to-day homeostatic function of a cell. Now, when these reactive oxygen species are produced in higher amount, they can create a pathological functions, for example, dysfunction of the cell or associated with disease. And how it happens, either superoxide um, automatically dismutates to hydrogen peroxide, or when there is a superoxide dismutase within the cell, which is antioxidant system, which makes hydrogen peroxide, and with iron freely available with Fenton reaction, it causes DNA damage, protein damage, and lots of damage to other macromolecules as well. And then it creates a pathological condition, what we most of the time see in inflammatory bowel disease or any inflammatory conditions of the gut or the mucosa. Now, coming back to NADPH oxidase. So NADPH oxidase is an enzyme which uses NADPH and uses this energy to make superoxide from oxygen. So it's a very simple enzyme in terms of chemistry. Now, what different with this enzyme is it's a membrane bound enzyme, and there are several isoforms of NADPH oxidase. So NADPH oxidase is a class or a family of enzyme, and there are different type of it. So one is NOX1, as I said, NADPH oxidase, in short we call it NOX. So NOX1, NOX2, NOX3, NOX4, NOX5, and there is another is DUOX. Uh, in general, what at the moment you have to just keep in mind that all are membrane bound enzymes. Some produces superoxide and some makes hydrogen peroxide as a reactive oxygen species. Now, depending on what type of ROS is produced will effectively determine what function this NADPH oxidase has. Also, where it is located in a cell, it is in a plasma membrane, phagosomes, or together with any other endocytic vesicles. Depending on that, its function will determine. But broadly, NADPH oxidase makes just two type of ROS, superoxide and hydrogen peroxide. Now, specifically, when we talk about the intestine, the NOX1, which is in here, is it the gut epithelium layer. NOX2 is a very famous NADPH oxidase. So any of you from background of immunology, you know that neutrophils and macrophages, uh, they act against bacteria. Now, how they act is they eat them, and within the phagosomes, there is an oxidative burst happening, and that oxidative burst kills pathogenic or even commensal bacteria, which is phagocytosed. And NOX2 is a critical component of oxidative burst, which is antimicrobial for those phagocytes, either neutrophils, macrophages, and up to certain extent, dendritic cells as well. So NOX2 is in the innate immune cells or phagocytes, what we call at neutrophils and macrophages. NOX4 is also in the gut epithelium as well as in the muscle layer attached to the basal membrane or the muscularis. Another NADPH oxidase, DUOX2, is also in the epithelium. Now at the moment, it just remember in a way that there are several NADPH oxidase in the gut, because when we talk about different functions, it will be much easier. Now, just to make visually clear, we have intestinal epithelium, we have DUOX2, we have NOX4, we have NOX1 in the epithelium layer. And when we say NOX2 in phagocytes, these are monocytes, macrophages, neutrophils, dendritic cells. They have NOX2, and their function is primarily antibacterial. They eat and kill pathogens or any bacteria they can see. NOX4 is also in the myofibroblast or smooth muscles. Now, this is the, in general, broadly what we see different types of NADPH oxidase in the host. But as we know that our gastrointestinal tissue is not only our tissue, sometimes we call our gut microbiota as a separate organ as well. So within that system, we have a commensal lactobacillus, and everybody has commensal lactobacillus. And these commensal lactobacillus, they also make a reactive oxygen species, which is hydrogen peroxide. I'm going to talk about more how hydrogen peroxide is secreted by these probiotic or commensal lactobacilli species, helps us in homeostasis or infections in later sites. But this is the overall how reactive oxygen or nitrogen species, for example, we have nitric oxide synthase also within the epithelium. So there are different ROS oxygen species interacting with RNS nitrogen species, and there is a complex interplay of these radicals um, and electrophiles. They go and interact with each other. So primarily we have superoxide, we have hydrogen peroxide. If we have nitric oxide in the vicinity, it makes peroxynitrate, which is very highly potent oxidative. It makes DNA and lipid damage. We have superoxide, which in the presence of myeloperoxidase from hydrogen peroxide makes hypochlorous acid. Hypochlorous acid is formed within the phagocyte vacuoles, phagolysosomes in the macrophages or neutrophils, which is highly potent in killing bacteria. And usually when this hydrogen peroxide is either antagonized by catalase, glutathione peroxidase, or GSH, or if it is made in a physiological concentration, it can act as a signal transduction secondary messenger, tissue repair, and is one of the important mediator of host microbe interactions. So this is very crude, but their interaction is much deep. So the meaning of me showing this is just for you to understand that reactive oxygen species is not standing alone. There are many ways it can interact. There are many molecules it can interact with, and that will determine what is the physiological function or the pathological function, that is disease coming out of those interactions. Now, this was introductions. Now, coming back to some of results and experiments. So this is the first results, particularly not done by me, but this was done in my current lab. And I wanted to show this because this will build up the story later on when I show different questions what we asked, and then it will be easy for you to understand. Now, the first and most important is, remember, as I said, there are different NADPH oxidases in the gut. So combinedly NOX1, NOX2, NOX3, NOX4. So what my colleagues, what they did is they made a genetic ablation or genetic modification in mice. So either they knock out, take out the NOX2, P22. Now remember, I'm gonna just show this here. So NOX1, NOX2, these are the main primary enzymes. Now, when we say P22, it's adaptor protein. It's a heterodimer, which is very important for function of all NADPH oxidase. So for example, when we make P22 knockouts, as you can see, none of the NADPH oxidase can work because it's an adaptor protein, accessory protein important for the function of NADPH oxidase. This is just a flux, so nothing to worry about. And this is This is P22 knockout, the red one in the whole body. In the right here is P22 knockout just in the epithelial layer. So when we knock out just in the epithelium, we won't see any NADPH oxidase working in just the gut epithelium, but anywhere else, it's all fine. Um, Now, why they made this thing is to ask a question whether NADPH oxidase made in the gut epithelium play a role in prevention against gut infection, because we know and we've seen that these from many other previous data that these reactive oxygen species, either superoxide or primarily hydrogen peroxide, they act as a microbial repellent. So they when they are secreted in the vicinity of the pathogen or microbiota, they changes their intracellular signaling of the microbiota and stops expression of their pathogenic proteins. So they act as a barrier, and they are repellent to these pathogens. So they are very important. Now, because NOx2, remember, it's in the macrophages and neutrophils, and this is important for killing pathogenic bacteria. So what they did is they made these different genetic modified animals, and they infected these animals with a pathogen called Citrobacter rodentium. Now, Citrobacter rodentium is same as E. coli, but for mice. So you can just understand this as a E. coli infection in humans. Now, obviously, in gray here, you will see, so this is a body weight. And there are many parameters taken out, but just for the simplicity, I'm just showing how the disease progresses based on the body weight. So as you can see here in gray, when you delete NOx2 from the animal, the body weight recovery through the body weight is slow, and dark black here is a wild type, which is a normal wild type. So as you can see, when you knock out the NOx2 knockout, the animals recover slower, and which was expected, because we know that NOx2 is preventive, it is antibacterial, and if we take that out, obviously the disease, it's going to take much more time to recover from the infection. So in here, which was expected. What was unexpected, because what they thought is same as NOx2 if we take out NADPH oxidase in the epithelium, because it's also the same ROS. It is preventive, it prevents from bacterial infection. We're gonna see exactly the same. But what they saw is totally opposite. So the red here you see is P22 knockout. So this is whole body knockout. So none of the NADPH oxidase are working, NOx1, NOx2, NOx3, NOx4. None of them are working, but still you see these animals, they recover much, much faster than the wild type. And the same when they knock out this P22 in just epithelium, intestinal epithelium, the same results. So this was very surprising for them, that having a ROS producing NADPH oxidase makes disease more worse in the epithelium, but quite opposite in the phagocytes, neutrophils, or macrophages. Now, there was something different, obviously, because this is not anybody expected. So they did so many things. One of them, what came out is, in here you see the white bar here is a wild type, and the black is P22 knockout, that is knockout from the whole body. And what they eventually found out that and this is at steady state, so there is no infection. So when they knocked out P22, when there is no NADPH oxidase working either in the body or the gut, you see increased abundance of lactobacillus, and more specifically lactobacillus reuteri, which is a commensal in mouse, and it is a probiotic lactobacilli family. And so at steady state, what they found is when you knock out these ROS production system, you have a huge upregulation of lactobacillus in the our gut microbiota, in mice microbiota. And they did a parallel experiment with Western diet, and they found that with Western diet, either wild type or P22 knockout, so again knocking out all NOx systems, there is no increase in lactobacillus. Now, this was surprising. Then again, even at homeostasis, just knocking out of NOx system, you have increased probiotic lactobacilli. And with certain more experimentation, what they found is um, this lactobacilli, and like many others, it makes hydrogen peroxide. So what they did eventually is, then obviously they made, and here I was part of it, making these um, knockouts of this lactobacillus, where because obviously Retri is a wild type commensal pathogen. So they got a different lactobacillus, probiotic lactobacillus, and we identified enzyme within the probiotic lactobacilli which is responsible for making hydrogen peroxide, and we deleted that enzyme. So what they did is they then infected these animals and then fed these to probiotics. And 533 is here, you see wild type lactobacilli, which makes hydrogen peroxide, and 9359 in here you see doesn't make hydrogen peroxide. And what these two bars indicate here is the expression of pathogenic island genes within this Citrobacter redention. What essentially it means is, when you feed mice, or the mice itself has a hydrogen peroxide producing lactobacillus, the expression of pathogenic island genes within this pathogen is downregulated. That means it is now less efficient and less potent to make infection to the host. So what eventually it means that you have hydrogen peroxide either being produced by epithelium or with the lactobacillus as at steady state. But because when you take out the Knox's system from the epithelium, there is a mutualistic relationship, and by default, to compensate this loss of ROS production from the epithelium, there is outgrowth of lactobacillus within the microbiota, and it can compensate with the production of hydrogen peroxide. So in here you see on the top, these top two panels, these are wild type animals, and these are P22 knockout, either whole body knockout or within the epithelium. So what happens is, when you have a normal wild type animal, there is an inner mucus layer. Most of the bacteria are at the outer mucus layer, and there is a superoxide and hydrogen peroxide production by the epithelium. And now, because we have knocked out the Knoxes, there is no ROS production by epithelium, but within the microbiota, Lactobacillus reuteri proliferate and overtakes the production of ROS, precisely hydrogen peroxide. So effectively within the system, there is not much change in ROS production. Now, when there is infection, this, the Citrobacter within the outer mucus layer is absolutely free, because there are just normal lactobacilli. The Citrobacter or the pathogen goes to epithelium, it starts its own program. Then after that, after some time, there is a recovery. But when there is a knockout of Knoxes, the Citrobacter, when it is at the outer mucus layer, itself it encounters the increased lactobacillus reuteri and hydrogen peroxide being produced by lactobacillus reuteri. So effectively there only it is encountered with reduced pathogenic island expression of genes, and which gives it less chance to go and interact with epithelium. So what it says is, there is a mutualistic relationship between our host and the microbiota, and one redox compensate with other redox systems as well. Now, this was published quite ago in 2016. And building on to this, so this is what I was talking about. So what we made is, we made we got a very well known probiotic Lactobacillus johnsoni. So the wild type lactobacilli makes hydrogen peroxide. We made a knockout of two genes within this lactobacilli, now which, as a result, this probiotic cannot make hydrogen peroxide. And at the same time, we we cloned this gene and then put it in plasmid, then transfected it again. So as a result, there is a version of lactobacillus johnsoni which makes more than normal hydrogen peroxide, which we call it overexpression, and which is then reflected in here, the bar graph. You'll see the blue one is the wild type lactobacilli, which is making a certain amount of hydrogen peroxide. The red one is overexpressor, which makes much more higher, and the green one is a deletion mutant, which cannot make hydrogen peroxide. So based on our previous observation, we asked a question that we already know that hydrogen peroxide is important for infection control. So we asked a question whether it is also important for inflammation. So uh, in mice, we have a model of DSS-induced colitis. So colitis is an inflammatory condition or inflammation of the gut, and it is a model in mice for replicating some of the traits of inflammatory bowel disease. So inflammatory bowel disease, again, a pathological condition in humans where either the large intestine or the small intestine is inflamed. So to model that disease in mice, we use several types of colitis model. One of it is DSS-induced colitis. So we generated DSS-induced colitis modeling and then used this probiotic to see whether hydrogen peroxide secreted by these probiotics have any role in epithelial restitution or proliferation or not. Because in the previous ones we saw infections, it was a direct link or direct interaction with the pathogen. Now we want to ask question: does this hydrogen peroxide has its role in the epithelium itself as a renewal or not, or because this is a ROS, this is gonna induce more oxidative damage. So just a summary of it, you see the blue ones here. Again, this is a body weight, so there is a DSS colitis, and you track animals over 14, 16 days, and you weigh them daily, and that will give you a fair idea how the disease is progressing. So when the red one you see here is DSS, so when you feed them DSS, there is a sharp decrease in body weight. So at day 8 and 9 is their peak of inflammation, and then after that they start slowly recovering. That's the model goes. Now the blue one here is a wild type 1x. So that means, for example, uh, these animals are fed with DSS as well as this hydrogen peroxide producing lactobacilli. As you can see, when they reach to their disease peak, once those fed with hydrogen peroxide producing lactobacilli, they start recovering much faster than the DSS itself or the lactobacilli, which is inefficient of making hydrogen peroxide. So effectively no change in that. So which clearly indicates that the hydrogen peroxide production plays a major role in recovery or the restitution, not the mounting or initial phase of the disease. And that can also be seen in here. So at day or 9, you can see this is a mouse colon. These are all damaged epithelium, thickened mucosa, lots and lots of infiltration. And when they are treated with lactobacilli, hydrogen peroxide, producing lactobacilli, hydrogen peroxide acting as a secondary messenger, initiates wound healing and renewal and replenishment of these epithelium. And when the same lactobacilli just knocking out two genes which are responsible for hydrogen peroxide, effectively there is no change, which clearly indicates that hydrogen peroxide, either in our case, yes, it is secreted by lactobacilli, but it can be secreted by adjacent epithelium cell, act as an important secondary messenger for wound healing and restitution. Now, the next question what we asked is, okay, so there is a certain amount of hydrogen peroxide produced. What will happen if we try to make, let's say, 10 times more hydrogen peroxide? So what will happen if there is more than normal hydrogen peroxide being produced? So this is a survival curve of similar experiment. So as you can see, in here, when I showed, there is an overexpression mutant, so which is making much higher hydrogen peroxide. So the red one is OE, so there is overexpression mutant fed together with the DSS-induced colitis, or a normal lactobacilli, but 10 times more concentration. And the green one is just the deletion with no hydrogen peroxide. And you can, as you can see, that within the same experimental setup, if we feed them either more of the bugs which are making hydrogen peroxide or just a normal bugs, but they are making lots and lots of hydrogen peroxide, the animals, the survival curve goes down. So the y-axis is survival. So the lower the number, that means more animals are dying over the course of experimentation. So which means that there is a certain limit and balance of ROS being produced, which can effectively be used for restitution and regeneration. But if that ROS production limit crosses a certain limit, it eventually backfires and can also aggravate more inflammation and more tissue damage. Now, we asked a question to the opposite extreme. What happens if we just quench all ROS? So if we just technically degrade all reactive oxidant species. So in similar way, I used a high antioxidant, which is higher than normal what we use. So either an acetylcysteine or a quinone, which is highly potent antioxidant. Within the same experimental setup, I gave DSS to induce colitis, and together with a high amount of antioxidants. And as you can see here, opposite to what normally many people see is that when you have lots of inflammation, you eat a lot of oxidant, you'll be healthy, and that is not going to work. Because when there is an inflammation, if you have a more than normal antioxidant, which is eating all up the ROS which are required for the restitution, there is no restitution. So it's also very bad. So eventually there is a balance, there is a moderate balance of ROS production, which is essentially very much important. If you increase that ROS, and there will be damage, or if you quench all ROS, if you eat up all ROS, then also it's not good for the host. And in general, this slide effectively shows the same way. If we have a certain minimal amount of ROS, it aids up the recovery. If there is no ROS, obviously there is no change and the recovery is slower. But when you have more ROS production, the recovery is slower as well. And because the ROS is damaging the tight junctions or the epithelial proteins, there is a chance of normal microbiota going inside the body and then making a sepsis, systemic infection. So this summary just tells you about that there are both roles of reactive oxygen species. It can be beneficial, it can be detrimental, depending on where, from what is it's being produced. When it is being produced, it's a constant. Where it is being produced, within the cell, out of the cell, what chemistry it carries, whether it's superoxide, hydrogen peroxide, and in what concentration it is produced, will determine eventually combinedly whether it is going to be beneficial or it is going to be detrimental. Okay, so now we are going to little more detail and little more complex. So now, until now, what we saw that there is epithelium and there are lactobacilli, there are host microbiota, which makes reactive oxygen species, which are important for pathogen control, as well as they're important for restitution. Now, in similar, what just remember I showed you, I told you that there is a pathological condition of the gut, which is called inflammatory bowel disease, and it's inflammation of the gut primarily. And there are two different types of IBD. One is Crohn's disease, and one is ulcerative colitis. In general, what just for now you can understand is Crohn's disease is patchy, so it can be in the large intestine as well as in the small intestine. Ulcerative colitis is primarily the larger intestine or the hind part of the large intestine. Now they combinedly are called inflammatory diseases, and it's called inflammatory bowel disease. Now it's a multi-etiological. Nobody clearly knows how it is triggered, but we have many factors which we think contribute to the development of IBD. One of them is susceptibility genes. So there are many genes which people have identified that if those patients have mutations or any missense mutations in those genes, they develop or they are more likely to develop inflammatory bowel disease. And one of those susceptibility genes are few NADPH oxidase. So for example, it's quite opposite to what we saw just before, that NADPH oxidase makes reactive oxygen species, and people thought that when you have inflammation, you have more ROS, you have more ROS damage. So when you don't have ROS, how can you be susceptible? There are certain theories that susceptibility comes up with because the body for a long time cannot tolerate microbiota, but in general that is what it is. So you have when you have a mutation in these genes, particularly NOX2 and NOX1, and they the patients, they are highly susceptible to IBD, and there are many IBD patients with mutations related to these NOX systems. Now, what is surprising is, for example, if we try to make animals, so if we just knock out these NOX2, NOX1, or P22, which is accessory protein of these NADPH oxidase, the animals are completely fine. There is no IBD, there is no susceptibility. So they are absolutely fine. There is no disease. So it's quite contrast what we think in humans are. Uh, in here, just for you to show what I'm going to talk about is two NOx systems. One is NOX1, which is a main protein NOX1, and there is a accessory protein P22, just for you to remember. And there is a NOX2, main protein NOX2, and then accessory protein P22. This is in the phagocytes, macrophages, neutrophils, and the NOX1 is in the epithelial cells. Now, in patients, there is one of a mutation which is associated with IBD, what they call is CYB and NMF333. So there is a mutation in CYB is a gene name of P22. So don't be confused. It's just a mutation in one of the accessory protein of the NOXes. And this mutation is a missense mutation, which leads to diminished activity of P22. So what it results is then eventually, of course, NOX1, NOX2 can't function. So in here you see, just for you to understand that when we have NOX1 knockout, because this is NOX1 is only in the epithelium ROS production is gone, but in the phagocyte, which is neutrophils and macrophages, they are completely fine. When we knock out CYBB, which is NOX2 again, and I'm repeating again because I know it might be a little complicated, but just for you to remember again and again. So when we have a NOX2 knockout, we have ROS production in the epithelium completely fine, but in the phagocytes with neutrophils, macrophages, they are gone. When we have a CYBA knockout, which is accessory protein for both NOXes, of course, there is no ROS production either in the epithelial cells or the phagocytes. And effectively, again, this is a body weight after DSS-induced colitis. And as you can see, the gray one here is just a wild type DSS, normal wild type. And you can see any other genetically modified animals, either P22 NOX1 or NOX2. They are not worse. Sometimes, which is NOX2, or it's better. They're recovering better. That's a different story. I'm not going to tell that about. But the most important take-home lesson is, and what we showed, what I told you just before, that even we delete all this, even after DSS-induced colitis, the animals behave as normal, as a wild-type DSS colitis. So their colitis is perfectly fine. They don't behave. They don't act like a susceptible to inflammatory bowel disease. Now, remember I showed there is a mutation, patient mutation. So what we did is we carried that mutation, made a P22 clone, and with this CRISPR, we made animals. And that was done by a different group, obviously, and this mouse is readily available for many studies. So this mouse has a mutation, which is a mutation in human inflammatory bowel disease. And you can see the red one when we do DSS colitis. Now, just because of this mutation, the animals are behaving very bad. So it's their body weight is they're losing body weight much faster, and this is just for hematoxylin eosin for you to understand that there is a certain inflammation in wild type, but these CYBA and MF333 mutants, they are much worse. So this is very surprising because when you take out the whole gene, the animals are completely fine. But when you have a certain specific mutations, which we know that it's not with that mutation, NOX1, NOX2 are not working. With just the mutation, but still the mutant protein is there, the animals are behaving really bad, and that reflects the susceptibility of IBD, and that is what we expect, that if any gene is is a susceptibility gene for IBD. This is how it's going to behave. But this was surprising that why it is working. So what we did is, so there is a technique called in vivo imaging system. So we inject certain chemicals in animals, and then we look for chemiluminescent signals, and that chemiluminescent signals comes out of reactions through to reactive oxygen species. So in here you see a wild type. So they have the red green is the bar here. So red is more signal, green and
और कितना
blue
मिनट
is less.
चलेगा?
बस थोड़ा है, ज्यादा नहीं.
ठीक है, ठीक है।
टाइम है, हो गया क्या?
हाँ, 50
5
मिनट
मिनट.
होने वाला है, तो याद दिला देना।
पता नहीं
क्योंकि
चला, ठीक
साइंटिस्ट
है.
तो बोलते रह जाते हैं, तो पूरा दिन उनको याद नहीं रहता है ना, तो बीच में याद मॉडरेटर का काम।
एक्चुअली
जी,
ये इम्पोर्टेंट है.
हाँ,
हाँ.
ठीक है
So
जी।
just I'm going to quickly short it. That's just
हाँ,
थोड़े
हाँ।
slides बचे हैं. So what here's summarizingly what I want to tell you is that when you have a CYB knockout, total knockout, you don't see any signal. That means there is no ROS production. But when you have a mutation, there is certain signal. And later on, we found out that this mutation can still make NOx4, which is another NOx, make hydrogen peroxide within the system. And that is why the animals are making just a little hydrogen peroxide. So in here, you can see when we have a total knockout, there is no ROS production. But when you have this particular mutations, in phagocyte, which is neutrophil macrophages, NOx2 is not working. But in epithelium, there is very little but important ROS production there. And because there is important ROS, really important ROS production from epithelial cells, strikingly, you see this is normal wild type, and this is homeostasis. In colon, you always have a thick mucus layer and you have thin mucus layer, and there is always a gap between the epithelium and the layer from where the microbiota colonization starts. And this gap is very important for our body or host to maintain a tolerance. But in these mutants, the thick mucus layer is gone, and the microbiota is actually encroaching to the crypt. So they are very close to the epithelial surface. And later on, we can actually quantify how much is the mucus thickness. And there are granule area, which is the goblet cells. So the goblet cells are making mucus, but they're not able to secrete it outside. And that's why the goblet cells are becoming bigger. So this reflects a susceptibility that this particular mutation causes these mucus layer to somehow change, and as a result this normal microbiota is now in close contact with the epithelial surface. And this is more or less the same with all mutants. uh, Everything is same behave as wild type except this NMF333 mutations. So later on what we proposed is there is a two-hit theory for susceptibility to inflammatory bowel disease. One is you should have no or very minimal ROS production in the phagocytes and very low ROS production, but production of ROS in the epithelial surface. And when these two factors are combined, then you might have a colitis. You means anybody or mice. That is what theory we suggest. Now this is the last slide, Gopalji. Don't worry. So remember I showed you here that there are certain changes in the mucus. So later on we identified that what changes is that the mucin production is completely fine between two animals, but there is a downregulation of a gene which is FUT2, which is an enzyme, which is a gene which modifies mucus. So it adds fucose residues to the mucus, which makes the mucus either thick or less thick. And there are certain glycosylation, certain lots of modification. Mucus is just not very simple protein. It's a lot of complex. And you can see the mucin production is similar, but in these mutants, UEA1 is used to see or visualize the fucosylation in the mucus. And you can see these mutants, they have less green, which is the fucosylation residues of the mucus, and their mucus is very thin as compared to wild type. So what it effectively supports the previous study that there is a deficiency in mucus structure, and based on that we did mass spectrometry and HPLC and identified what core residues are different in the mucus, eventually just stating that the lack of ROS production effectively alters the mucus constituents, and effectively then leave the area open to the commensal microbiota to encroach and get closer to the epithelium, and as a result then it is more susceptible to have a colitis or inflammation. That's all. Thank you.